Hello and welcome to today's presentation about nanoscale sensor networks, or how DNA can be a new paradigm for communication at the bottom. My name is Dr. Florian Lennart-Lau and I am from the University of Lübeck from the Institut of Telematics. But without further ado, let us have a short overview about today's presentation. I will begin with a short introduction of my research group and myself, and I will be explaining what we do. I will further motivate the general branch of research of nanonetworks, or DNA nanonetworks in particular, and present a few applications as well as the state of the art, and make a short prediction about the future growth of the research topic. Afterwards, I present the biggest unsolved problems and define all the necessary terms at the nanoscale, as well as some basics concerning DNA. After that, we dig into the main content of the presentation and present several message molecules and several DNA-based nanonetworks. The novelty, or the paradigm shift this method presents, is that we compute inside of messages and not inside of computational devices as we are used to at the macro scale. And after that, I present a short conclusion and a summary. Okay, but let us begin with a short introduction of myself. I was born in 1990 in Lübeck and finished my education in the year 2009. After that, I finished my bachelor's degree on peer-to-peer systems in 2013 and my master's degree in 2016. I switched topics to theoretical computer science and wrote a thesis on the competitive analysis of algorithms. After that, I started working as a research assistant under Professor Dr. Stefan Fischer and started conceptualizing DNA-based nanonetworks. In 2020, I finished my thesis with magna cum laude, and since then I'm working as a postdoctoral researcher at the Institut of Telematics at the University of Lübeck. There I'm currently organizing the workshop WONAN, and I am the author of a lecture on nanonetworks and a seminar on nanonetworks, and currently writing all kinds of journal and conference papers on the topic. I am part of the Nano Group, which is our research group. It was founded in 2017. And the members are myself, Mark Stelzner, Regine Wendt, and at the bottom right, Professor Dr. Stefan Fischer. And we focus on all kinds of different topics concerning the nanoscale and nanonetworks, beginning with nanocomputation, DNA-based nanonetworks, and other things like ethical, legal, and social consequences as well as constraints. Further, we obviously focus on protocols and all kinds of algorithms concerning networking algorithms and engineering aspects, as well as simulation aspects, and many more topics we will discuss in more detail in this mind map. As you can see here, we have created a mind map of all the topics we are currently researching on. Among those are evaluation mechanisms like simulations, components of nanonetworks in general, like locomotion components, actuators, sensors, energy harvesting mechanisms, ethical, legal, and social constraints, computational aspects like quantum dot cellular automata, chemical reaction networks, self-assembly systems, CMOS technologies, and all kinds of other things. Further, we research all kinds of applications for nanonetworks. Among those are treatment mechanisms for things like pulmonary infections and other things, as well as environmental factors and biodegradability, waste disposal, and those things. And further, we research some formal constraints about nanonetworks, like machine models. We define things, we derive parameters for simulations, and lastly, obviously, we research the networking aspect, the communication aspect, and the different kinds of communication that might be feasible at the nanoscale. For example, electromagnetic communication in the terahertz band, or molecular communication as is presented in this talk, or maybe even acoustic communication or other kinds like Coulomb, electron interactions and other things. Okay, but let us now come to a short motivation on the entire topic. Well, First of all, where does the word nano even come from? It is an ancient Greek word, and nanos means dwarf or something very small. And the first mentioning of the word atom, the smallest particle by definition, occurred in 500 years before the Common Era. And other countries and other cultures came up with similar concepts. For example, the concept of the Kalapa surfaced around 600 years after the Common Era in India. And in general, we define everything to be nanoscale if it is the size of 1 to 4,000 nanometers. Technically, only everything between 1 and 999 nanometer would be fit to be called nanoscale, but since the smallest human blood vessels, the capillaries, are about that size, we figured that we should include everything that can fit into those vessels, as most people would call human blood cells, red blood cells, something along that line too. And as a comparison, human hair, for example, is between 20,000 to 200,000 nanometers in diameter. And a cell is 10,000 nanometers, and the DNA helix, also for comparison, is only 2 nanometers in diameter. And to have a more relatable example, you could compare the nanoscale when we look at the size of the Earth compared to a soccer ball. And the same ratio is also true for a soccer ball compared to a carbon nanotube. And to the right, we can also see the hair comparison. And the background, the very thick looking structure is a hair. And in the front, all the tiny fibers are nanofibers. So they are much, much smaller than all the other hair structures in the human body. And here is a more vivid example even. What we can see here is a size scale ranging from 10 to the power of -1 to 10 to the power of 8 nanometers in size. To the right of the scale, there are tennis balls that are roughly 10 centimeters in size. And if we go down further down the line, between 1 to 10 to the power of 2 nanometers, there are all the interesting structures at the nanoscale. For example, proteins, DNA molecules, viruses, sometimes bacteria, and also structures that have been proposed as transport mechanisms for medication, like liposomes or gold nanoshells or even quantum dots, and all kinds of other structures. All those things are potential building blocks or materials we could utilize at the nanoscale. But today we will focus on DNA as one of the main components of interest that can be used for all kinds of purposes. And per definition, DNA is nanometer sized. And there are all kinds of use cases even before the modern scientific era. For example, the German sword Ulfbert has already been reinforced using nanotechnologies without the knowledge of the blacksmiths involved. The sword has been created between the year 800 or 1100 before the Common Era, and it is made of carbon reinforced steels. And the carbon source for this sword was bones. They added bones of killed animals, for example, to the forging process, and the carbon in the bones has reinforced the steel. During that time, there was a misbelief that the strength of the animal or enemy passed from the bones into the sword, but nonetheless, the sword became stronger. And there are all kinds of other applications that we will now analyze. For example, the most commonly named application would be medical surveillance of health parameters. People envision that we can add a swarm or a network of nano devices to the human bloodstream, for example, and use it as a monitoring device for health parameters. Those nano devices might even be able to fight diseases or illnesses when they are still in the process of emerging without any macro scale or systemic symptoms we usually use to diagnose a sickness. Other applications lie in the area of the military, where people envision nanotechnologies to be used to monitor communication processes. But all kinds of other applications are possible too, like agricultural applications, where we could imagine that nanotechnologies could raise the efficiency through, for example, increasing the efficiency of distrains, like breaking down organic tissues to make nutrients available in the soil. And a similar process could be or a similar result could be achieved by improving the efficiency of rhizome networks that are oftentimes symbiotic with all kinds of other organisms in the soil. And lastly, nanonetworks could obviously assist in the prevention of disease or in the control of disease in the soil. And the agricultural aspect is of rising interest, because we can't really test many of the nanoscale networks or nano applications inside of living organisms for ethical reasons. Thus, it makes much more sense to test them in other environments first. And among those are also environmental control or regeneration tasks, or even early warning systems for natural catastrophes like tsunamis or earthquakes. Sometimes it might not be possible to use macro-scale sensors, for example, and add them to the soil to detect earthquakes early on. Okay, but let us now come to a more schematic view of the entire process. What we see here is a schematic representation of a human body. And we divided, or Stefan Fischer divided, a nanonetwork or a body area network into 3 components: one nanoscale component, one microscale component, and one macroscale component. All those components communicate with each other. The tiniest component in this example would be the small red dots that are distributed around the body of this person. This is a nanonetwork. The nanonetwork itself collects, for example, medical data of a patient and communicates it to a gateway which is marked in red. During that process, nanoscale communication processes are used, for example, molecular communication or terahertz communication. And the results can be communicated to implantable devices like insulin pumps or pacemakers or anything like that, to make better judgment calls on when to activate those devices. And if that's necessary, the results of all those measurements can also be communicated to a macro-scale gateway, where the collected data can be sent to other remote devices like laptops or sent to doctors for further judgment. And this is more or less the scenario we envision when we talk about nanoscale technologies. Obviously, it is not possible as of yet, but it might be in the quite near future, maybe 10 to 20 years. Okay, and another scenario we analyzed in more detail is the detection and maybe even treatment of pulmonary inflammations. And the discovery of such a disease could happen via markers, via a body area network. And the communication of the discoveries to other devices can happen via a gateway, for example, as already displayed in the schematic representation of the human in the last figure. And it is also possible, or it might also be possible, to directly treat the disease, the inflammation, via the process of emergence. When the human immune system can't keep the inflammatory component at bay, we might be able to assist with nanotechnologies. And one of the major advantages of those nanonetworks is that we can use local treatments without damaging the surrounding tissue. And this is a yet unsolved problem in medicine. Right now, if we want to combat, for example, an infection, we most of the time apply some medicine systemically. It acts in the entire body and has lots of side effects. With nanonetworks, it might be possible to limit the release of medication to just the area that is affected by a disease. In the case of a pulmonary inflammation, it might be sufficient to just release antibiotics in the lung and nowhere else. That way we might be able to avoid side effects in the gut biome or things like that, which would be a major breakthrough and a yet unsolved problem in medicine. But let us now come to the state of the art in nanotechnology. Right now, there are lots of disciplines involved in the nanonetworking community. It is a strictly interdisciplinary field of research, and all kinds of disciplines add knowledge to the entire sector. First of all, the biggest influence we know of is the sensor networking community with all the protocols and algorithms involved. But also, mathematicians add all kinds of analytical tool and simulation tools to the equation we can use to simulate the diverse environment at the nanoscale. Those are much more versatile and much more different from what we are used to at the macro scale. Further, medicine, for example, supplies us with lots of different use cases and clinical trials, as well as pharmacy with exploring possible kinds of medication that might be compatible with nanobots or nanonetworks in general. And bioengineering pioneers all the components that we might use for construction processes. And lastly, biology, chemistry, and physics explore all the processes that might be involved in the construction of nano devices. It is not yet clear how those nano devices might be constructed. It might be due to top-down approaches or to bottom-up approaches using self-assembly. The latter will be explored in today's presentation. But what are the existing nanostructures that we can already create in laboratories? First of all, nanoparticles for disease control or, for example, for fighting cancer are already part of clinical trials. People already use them to fight cancer. Further, it is already possible to create primitive microbots that can, for example, be swallowed as a pill and monitor certain values in the gut biome. It is also possible to already create certain transport structures like liposomes or DNA boxes, or artificially engineered dead bacteria that serve as a carrier vessel. And lastly, it is also possible to create all kinds of DNA-based nanostructures using the process of DNA origami, where a long DNA strand is folded to create a desired shape. And just for the sake of illustration, we can use, for example, gold nanoparticles to fight cancer. And this happens by coating the nanoparticles with antibodies, with specific antibodies. By doing that, the nanoparticles can enter certain cells or certain tissues where the cancer resides. And by heating the nanoparticles externally via lasers, they start vibrating and heating up, and that process of heating up the nanoparticles can kill cancer tissues, while the healthy tissue, the healthy surrounding tissue, is able to regenerate from the damage. And just as a small illustration, there are aluminum nanoparticles displayed in the bottom figure. Further, it is also possible to create certain imaging algorithms using nanoparticles. For this process, we can use iron oxide particles the size of about 10 nanometers. Those can assist in visualizing biological processes. And they are activated by externally applying magnetic fields, and by calculating from the resonance we can find out where certain things are situated in the human body, and such processes can, for example, be used in MRT and other imaging methods. And this is how we envision certain types of electromagnetic nanorobots. This is obviously just a schematic drawing, but we can see all the components we regard as necessary when it comes to nanorobots. At the top, we see a plasmonic graphene antenna that is used for communication processes. As we know, in classical sensor networks, the majority of the energy consumption of such a network happens by communicating. Thus, we need a especially big battery at the nanoscale, and we use zinc oxide wires for that purpose. And the zinc oxide wires use the majority of the space in this entire nano device, as we use lots of energy for communication processes. Further, all kinds of processing components like memory, a timing mechanism, and a processor are necessary to collect and process data on the nano node or nano device itself. The timing component is necessary to put a timestamp on the measured data, otherwise it is pretty meaningless. If we can't tell when an event occurred, we can't really use the data. And further, we have actuatory components and sensor components that can act and react to environmental variables, and maybe a propulsion motor L that can be used to propel the nano device or micro device to certain locations in the body. This is how we envision the entire devices, but as of right now, it is not possible to manufacture those. Another option that might be more feasible right now is modifying already existing structures at the nanoscale. For this purpose, we use, for example, cells. cells already contain most of the components we need to realize nanorobots. For example, the flagellum, or the molecular motor as it is sometimes called, could be repurposed for artificial locomotion mechanisms, and the natural receptors and gap junctions could be used to detect certain environmental parameters and act via releasing medication or other payload. And even those mitochondria could be used to manufacture energy for the entire cell, and for example the DNA or other components of the cell could be used for computational or memory purposes. And a very similar approach would be this bacteria-based nanorobot. It is pretty much the same idea as in the cell, but those bacteria bots might also be able to move along a concentration gradient via the process of chemotaxis, where they detect certain environmental parameters like a concentration of a specific substance and move along the gradient towards the target area, which might not be possible with cells. And bacteria has appropriate sensors and actuatory units that it can use to maneuver its environment. And further, the entire body of the bacteria might be usable as a medical payload transportation device, while the DNA might function as a memory unit. And it is even possible via the process of DNA origami or the process of tile-based self-assembly, DNA tile-based self-assembly, to create all the structures that are displayed in this picture here. It is from 2015, and as you can see here, there are all kinds of boxes, DNA-based smiley faces, stars, and all kinds of interesting symmetric structures, and even three-dimensional structures, as you can see in the bottom two rows, that can already be manufactured at the nanoscale. There's huge potential using this technology, and there's quite a bit of research going into this area. And we can expect further growth from the construction processes that are already available. DNA origami and tile-based self-assembly have a huge potential to assist in constructing certain objects or structures at the nanoscale. But let us now come to a prediction about the future growth of the entire research field. What we can see here is Maslow's hierarchy of needs. Those are all the human needs that are more or less involved in our day-to-day living. First of all, we have to cover all our physiological needs, our safety needs, and our social needs, and maybe even our individual needs, before we can think about self-actualization. And as soon as we fall sick or ill, it is our number one priority to fix that problem. And when we have a look at the distribution of the US GDP, we see that health is among the top concerns of all, and it is the most important aspect of the Maslow hierarchy of needs. When we combine those two facts, we can deduce that we expect the health sector to further grow, and since there isn't much new frontiers, a lot of the funding flows into possible nanotechnologies. And we expect that to grow, and our entire university kind of specialized on medical applications, as we see a lot of growth potential in the entire sector. And nanonetworks as a whole, and especially DNA-based nanonetworks, have a huge potential on that part. And when we think about all the available nano-networking technologies, we have a few very general unsolved problems. First of all, the construction of nanoscale functional components for nano devices or even networks of nano devices is of yet unsolved. We have no idea how to properly construct those devices. We have some concepts and some ideas, but those are rather abstract. And as of right now, to the best of my knowledge, we have not manufactured many or any nano networks or even nano devices that are comparable to the complexity of macroscopic devices. Second, the communication or communication mechanisms at the nanoscale are object to heavy research. Most people focus on terahertz communication, but during the last few years, the process of molecular communication, aka using actual particles, became much more prominent and the object of a lot of research. And in today's presentation, we analyze some implementation of molecular communication using DNA molecules, special DNA molecules, as we will see. And third, the computational aspect is also object to lots of restrictions and constraints at the nanoscale, and we are not yet sure how to implement those. While classical circuits can be already the size of a few nanometers, for example, the required batteries would be 1,000 times as big, since those circuits require a lot of energy. And lastly, and most importantly, the compatibility of all those sub-solutions is very much object of research. When we use the same component, for example DNA, to create all those components, we don't really have to think about the compatibility. But all the detailed partial solutions, as people suggest, are usually of unclear compatibility. Okay, but let us now come to an analysis of the nanoscale in general and what is already possible, what are the peculiarities, etc. Okay, there are a few unique points that we have to address at the nanoscale. First of all, each nano device has a very tiny communication distance, and thus we often need an extreme number of participants, even up to the million devices, to just have the ability to communicate through the entire human body, which is quite challenging, since communicating via through tissue or blood has quite a short distance. Further, if we apply those nano devices in the human bloodstream, there is a very high topology dynamic, since the human blood is obviously moving in circles, and we can't really tell where the nanobots are situated at any given time. Further, due to the very tiny size of all the devices, we have time and space restrictions. Those devices might not be capable of computing everything our macroscopic devices can. Further, those networks are quite heterogenic, and there is no real central control instance, and we can't really exert any steering influence on the entire network once it is applied. It has to function fully autonomously. And it should be biocompatible if we want to apply it in a human body. And it should function using some available kind of energy, as we certainly have some type of battery problem. We have to maybe harvest environmental energy to power and fuel all those devices. And lastly, it is unclear how to deal with the waste products of such a network if we apply it in a living organism. Those are just a selection of all the problems we might encounter at the nanoscale. Okay, but let us now analyze what kinds of devices we are talking about. During the process of this presentation, we are mainly interested in first nanostructures, which would be all kinds of artificially created nanoscale structures, nanodevices, which is an umbrella term for all kinds of nanodevices that at least possess some kind of energy harvesting or energy supply mechanism. And then there are nanosensors that we use to detect certain markers, which also have a sensory component and a component that can process information. Then we are also interested in nanonodes, as they can communicate results over multi-hop networks, and nanomachines which can act. They can, for example, release medical payload. They have an energy component and an information processing component, and a component that can activate, for example, a switch and open a box or something like that. And in the middle, we have nanorobots, which possess all those components and are the most sophisticated type of device we have. Okay, but let us now come to a motivating scenario that I have created. What we can see here is a person with potentially multiple ailments, multiple problems. In today's modern environment, this person might have to be treated stationary in a hospital. But as an alternative, we might be able to inject some kind of nano network into the bloodstream of this person. Then this person might be able to follow their daily abidings quite normally, and the nano network inside the body monitors all the health parameters of the person and communicates the results to a remote location where a doctor can make judgment calls about the health status of the patient. In some predefined cases, it might even be possible to directly react to certain changes in health parameters. But let us now have a look at the nano network that we injected into this person in more detail. It is a DNA-based nano network. What we can see here is a network of nano devices or even nanosensors that are able to detect certain environmental parameters represented by the orange rhomboids. Once those devices detected those environmental parameters, they release their payload, and that payload might be building blocks of a message molecule. And once enough of those building blocks are present, the message can fully form and circulate inside the communication medium, for example, the bloodstream. Once another nano device detects those messages, they can react to the detection by either releasing medical payload or by informing us of the detection of a certain disease. And this process is the basis of the networks we are analyzing today. It is a very different kind of networking we are used to as computer scientists, but it is interesting nonetheless, as those networks are quite powerful, and they can be created from already existing components. And the building blocks for the messages could be something like DNA tiles, as we can see them in this picture. What we can see here is a specific DNA molecule, and this is a class of DNA molecules that we can manufacture in laboratories. There are 4 DNA strands that are intertwined according to the base pairings. What we can see here is the base pairs adenine and thymine and guanine and cytosine, and they can only form a stable binding with their respective counterparts. Thus, we can encode certain binding conditions in those DNA strands. And these tiles are quite stable. And all of them in any direction can have an open strand that is able to bind with other DNA tiles and form a message molecule, for example. And since we can choose the basis of this open strand quite freely, we can encode a condition for a construction process. And when we choose those conditions smart, we can quite literally program the assembly process of matter. If we choose the appropriate conditions, as we will see in further examples, we can make sure that only a desired structure will grow if enough of those tiles are present. Those are the basic building blocks, the atomic components of message molecules. But let us now come to a short formal definition to understand the process in more detail. Formally, or mathematically speaking, DNA tiles are non-rotatable blocks as we see to the right. Those blocks have glues at each side, potentially on each side, and the strength of a glue is represented by a number of black boxes. 2 black boxes are a glue of strength 2 and 1 black box is a glue of strength 1. Further, those glues are implemented by open DNA strands, and thus they have an encoded base sequence that is represented by a label. I just called them A, B, C, and D. And this is a condition that is encoded in the open DNA strand. And only other tiles with a complementary basis can form a stable binding with other tiles of this type. And there is also a marker in the middle that can be used to interpret the results of computations, because these molecules are Turing complete and can compute any mathematical function a regular computer can perform. And here are some important properties of those DNA-based tile assembly systems. Tiles can compute, they are Turing complete. They can further construct as they take up physical space and as the binding process, the interaction of those tiles, performs a computation. It also performs a growing process at the same time. And tiles can be used to communicate in networks by simply encoding a binding condition that is representing some address of a nanorobot. And once a message molecule, a tile in this case, passes by a nanorobot with an appropriate receptor, it will form a stable binding with it and thus communicate a message, for example, that could be encoded in the tile. And lastly, all those tiles are influenced by the temperature, by the physical temperature of our medium. When the temperature is high, the bindings break up and the whole system is very dynamic. And when the temperatures cool down, the whole process gets slower and crystals form and message molecules form. And if we choose the temperature right, the message molecules will form. And we can see the whole process in some example to the right. The entire assembly process starts with a seed tile that is usually called sigma. This is a temperature 2 system, which means that we need a glue of strength 2 to form a stable binding. Otherwise, the temperature is too high and it can't stably bind. So we need 2 black boxes. And for example, this other tile B is an appropriate candidate to form a stable binding with the sigma tile. It is called B, and it has a fitting glue with 2 black boxes and the same label with the complementary bases. So it can form a stable binding. And the same is also true for this tile 1, which has a glue of strength 2 with the label B on the right side. And now we have an interesting condition, because the glues for a stable binding can also be supplied by 2 different neighbors, namely the tile sigma to the top right and the tile 1 to the bottom left. Now this tile, the other tile 1, can bind here stably, fulfilling the condition that the glue strength 2 has to be met by supplying 1 glue to the top right and 1 glue to the bottom left with appropriate labels of 0 and 1. And this is a mechanism, the condition we use to implement or to prove the Turing completeness of these tile assembly systems. And in terms of DNA, the whole process looks something like this. We already have some structure to the bottom right that consists of intertwined DNA strands, and there is a certain condition via the open DNA strand. And only another DNA strand with the appropriate complementary bases can form a stable binding with the already existing structure. And in this case, the TACG base sequence can form a binding with the ATCG sequence. And to the bottom, it is also a fitting sequence. Thus, the entire structure can form a stable binding with each other. And we create a DNA assembly of 4 tiles. And the whole process is also available in 3 dimensions by simply adding glues to the top and to the back side of a tile. And thus we have something that resembles a 3-dimensional coordinate system. And those things can also be used for constructional purposes. And in 3 dimensions, the cubes that I already spoiled a bit can already be created using DNA origami processes. And it is quite interesting because we can use them as nano robots or nano devices that can open themselves and release their payload upon detecting a certain DNA sequence. And those things have already been created in laboratories in 2009. And the mechanism behind the DNA box is quite simple. The lid of the box binds with the bottom part of the box with a relatively weak binding. When a specific DNA marker molecule is also present in the medium, it can form a binding with the top part of the box. And when that happens, they bind stronger with the box than the bottom part, and the box opens up, and we can release either tiles for computational or message molecule assembly purposes, or for the direct treatment of a disease. But it is often better to first release tiles and make certain computations on the input data to ensure that a treatment is really necessary. Upon the detection of just one marker molecule, there are sometimes errors to be expected. Okay, but let us now come to an interim conclusion. Thus far, we mainly showed what is possible and what can be constructed, but we have talked little about computational and communication processes. But let us change that now. With DNA-based nanonetworks or message molecules, it is possible to compute inside of messages instead of nanorobots. We thereby revolutionize the paradigm for computation at the bottom, and the computations can happen via conditional message assemblies. And only when a mathematical function is computed, the message molecule can finish and the computation result can be communicated. And the input for those computation can be released by such boxes that I just presented. And once they successfully computed their function, they form a receptor or a ligand, and now the result of the computation can be communicated. And the advantage of that approach is that it can work at the nanoscale as of right now, and there are much fewer constraints in the communication channel compared to inside of nano devices where it is unclear how much resources are available. Okay, but let us now come to an example in message computation. What we see to the left is a tile set of the computation of a 4-bit AND mathematical function or decision problem. There are 4 bits, the bit 1, 2, 3, and 4 marked in red, and a lot of other tiles that are necessary for the creation of a message molecule. And the entire process starts again with a seed tile sigma. Without the seed tile, nothing can happen. Once the seed tile is present, the assembly of the entire message molecule can begin. This is a temperature 2 system, and there are 3 possible locations for other tiles to form a stable binding according to the conditions encoded in the glues. To the top, to the bottom, and the first input tile for the computation of the end to the left. Once it is present, we now have other different spots where a binding can happen. For example, a top framework tile can attach and a bottom framework tile can attach to ensure the stability and the condition that a receptor may only form once all the conditional tiles, once all the input tiles for the 4-bit AND are present. And we can continue this process by adding more of the framework tiles and more of the AND tiles until this molecule is nearly finished. And now we see why we need the other tiles at the border. Now we can ensure that all the other conditional tiles are there before a receptor can form and computation is regarded as successful. As we can see here, now a receptor at the top and a receptor at the bottom can form. And those have open glues to the top and to the bottom. And if there is any other nano device consisting of DNA that has appropriate glues with the right spacing, they can detect this message molecule, and they then know that a 4-bit AND on a predefined set of input data has been performed successfully. And if such things happen, we can be quite sure that a lot of devices, in this case 4 different types of devices, have detected a certain event. And if that event is a disease, we can be quite sure that it is there and it is not a false alarm. They compute a distributed consensus among different nano devices. And again, the full assembly can only happen when all tiles 1 to 4 are present. Otherwise, this message molecule cannot finish. And we can now have a look at the entire network from a network perspective and not from a message molecule perspective. What we can see here are nanosensors 1, 2, 3, and 4. Those represent types of nanosensors. If all of those detect certain markers, they all release their tile payload, and those are the tiles 1, 2, 3, and 4. And all the other tiles required for the assembly of such a message molecule are already present in the communication channel. And once all of them are present, the message molecule can form, and thus the successful computation of the 4-bit AND, or the distributed consensus, is signaled. And those messages can be detected by nanorobots, and they can react by either releasing medical payload or by signaling a detection by fluorescence markers or something like that. And the entire process can be further modularized into 5 different phases. First, the marker detection phase, where nanosensors recognize the presence of a disease marker. Second, the tile release phase, where the nanosensors release their payload and maybe even repeat the detected signal to other devices. Then the message assembly process, the receptor binding, and the medication release phase. All those phases can already be tested in wet lab experiments. For example, Lee et al. Have already proven that the detection of an arbitrary marker via a nanosensor is possible, and that we can release medical payload upon detection of certain markers. The storage of tiles or drugs inside of nanostructures is also possible and has also been proven by Lee et al. And Anderson in 2009. Further, the assembly of message molecules in the laboratory is also possible and has already been shown by Lee et al. In 2004. And the attachment of message molecules to a nanorobot has already been shown in 2009. And lastly, the point 5, the release of tiles or drugs, is the same as point 1 and has already been shown. To the best of my knowledge, there's just no combination of all those phases that have been proven in wet lab experiments. Okay, but let us now have a look at a more complicated function. Thus far, the AND was a pretty simple mathematical operation, but it is also possible to compute functional problems where the answer or the output is not just a single bit, the information yes or no, but the sum of numbers, for example. And the behavior of an add or an addition is quite simple in binary. We regard the two input bits and the carryover bit from the computation if there was an additional number. And according to all those possible cases, those 8 cases, a specific tile has to bind with an assembly. And we can see the entire process in this example. In this case, the top tiles you can see now are the algorithm that performs the addition. And the whole process starts with a seed tile again, but this time we have a temperature 3 system. The only reason for that is that the temperature 3 addition message molecule looks a lot like a binary adder and thus is relatable and easily understandable by computer science folks. But in this case, we also create a framework structure, but now an entire binary number is released by nanosensors and binds to the top and to the bottom part. And now the glues, the open glues in the middle, encode the binary information of 0 and 1, and only one specific tile of the algorithm can form a binding here, according to the carry bit and to the 2 input bits. And once the process finishes, we can see that the sum of those 2 binary numbers is created in the middle, and only when that happens can a receptor form on this message molecule, and the computation is regarded as successful by the nano network. And this is quite interesting. In this case, the nanosensors release either parts or entire binary numbers, and they can be added by the algorithm that is already present in the medium. And once that happens, the message molecules are fully assembled and can be detected by nanorobots, and they can react appropriately by releasing, well, whatever they want to release. And it is even possible to compute more interesting functions like thresholds. Thresholds are especially interesting because many diseases are only significant when a certain number of markers are present in the medium. And in this case, a threshold can be implemented, similar to an AND message molecule. We have to release or create certain types of message molecules for all the possibilities of creating a threshold of 3 of a certain number of input bits in this case. And the threshold of size k can thus be implemented. And all the message molecules that are created can intuitively be regarded as different versions of the AND message molecule. And we see an example in this case. We have 4 input bits. We can see here the input bit 1 and 0 and 2 and 3. And if we want to create all possible message molecules that implement a threshold of 3, we have to create 4 of those message molecules. And as a nano network, the whole process looks something like this. The nano sensors detect certain parameters and release the respective conditional tiles, the input tiles, for the assembly of the specific threshold message molecules. And it doesn't matter what of those message molecules finishes first. As soon as the threshold is met, the nanobots at the bottom can detect the message molecules and the computation is regarded as successful. And thus we can implement such a very useful function as a threshold in nano networks. The number of message molecules sadly scales very badly as we have to compute the n choose k mathematical operation, which can result in quite a number of message molecules. Okay, but it is even possible to generalize the entire procedure and compute arbitrary Boolean formulas. For this process to function, we obviously have to convert our computational problem into a Boolean formula if that is possible. In that case, we then can create a truth table and compute the disjunctive normal form of our formula phi. And then we can create a message molecule for each of the clauses of the truth table. And the Boolean formula is successfully evaluated once a single message molecule has fully assembled. And we now have a look at an example. What we can see here is an arbitrary truth table, and we look at all the lines with a 1 in the last column of this disjunctive normal form. all those lines with a 1 are message molecules we have to create. And all the formulas or sub-formulas, phi1, phi2, etc., are the inputs to the computation. This special type of representation of a Boolean formula works just on the input bits, computes an end on all the inputs, and then there is a big OR that combines all the clauses in case you do not know what a disjunctive normal form is. And then for each of those rows of the table, we create a message molecule that represents all the inputs with the appropriate truth values. And this is pretty much the same as creating an AND message molecule or as creating a threshold message molecule. And for each of the lines, we create 1. And then if the appropriate tiles are released, we can perform the computation that was encoded in the Boolean formula. And the entire process is visualized in this nano network, where nanosensors 1 to nanosensors n release their respective tiles and perform the computation in the middle. And again, it is sufficient for a single type of message molecule to fully assemble for the computation to be regarded as finished. And then nanobots can react to the detection. And it is even possible to compute arbitrary mathematical functions using a simulation of a Turing machine. And I will only present a proof sketch, which is only theoretically of interest, as the resulting message molecules are quite big. All of us probably know Turing machines. They consist of tapes and head positions and state automatas, and we can simulate the entire configuration graph history of a Turing machine using a self-assembly system. AKA, we take the numbers on the tape and represent them as tiles, and we take the head positions of the Turing machine and represent them as tiles, and we represent the entire program, the automata of the Turing machine, also as tiles, which is possible. And we recreate the state history of the Turing machine using a stack of rows of tiles. And yeah, that is pretty much the process. For example, if we look at something like this, to the top, we see the tape of a Turing machine in the initial state, then in state 1, then in state 2, and then in the final state where the computation is over. And we can recreate these tape states using rows and stacks of tiles, as we can see at the bottom. And thus we can finish our computation the same way the Turing machine would, which is quite intuitively possible. And as a network, it might look like this. Again, we have all our nanosensors that release all the required tiles for the Turing machine simulation, and then the message can form. And in the end, there can be a receptor that can be detected by other nanorobots. It is possible, but it takes up a lot of space, and it's mainly of interest for theoretical purposes. And lastly, here we have a table of efficiently solvable problems by the Boolean formula approach, which is somewhat feasible. Truth tables are also growing exponentially, but for small input sizes it is definitely possible, and those might be enough for many of the medical scenarios that have been suggested thus far. And what we can see here is a list of pretty easy problems that have been suggested as parts of a computation in medical problems. All those problems have been extracted from certain publications that suggested to treat a certain disease using nano networks. And there are all kinds of other problems that lie in the same classes. We know that from complexity theory, for example, that can also be solved by those kinds of devices. And intuitively, those are rather easy problems. And all those problems can be computed by the Boolean formula approach that I have just presented. In theory, obviously, those networks can compute anything by the Turing approach that I have just presented, but there is a lot of overhead involved and a lot of errors can be the result. And since there might be size restrictions, it might be better to use the Boolean formula approach that I have presented, since there are already all kinds of functions that are possible that we can compute. Okay, and in summary, there is a number of problems that we can now solve that have previously been impossible to solve. Those are problems in the medical context that are very real, for which no real sufficient solution exists of today. And one of those problems is the continuous medical monitoring without impairing the quality of life of the patient. Currently, that is not possible without a stationary environment or laboratory conditions under considerable deprivations of life quality. But if we inject a nanonetwork into a person, it might be possible. Further, the immediate treatment during the emergence of a disease is also not possible right now, but it might be possible using DNA technologies as those that I have just presented. And lastly, the local treatment without systemic consequences is thus far also not possible, but it might be possible using such nanonetworks. They can react immediately upon detection of a certain type of disease marker, can compute a distributed consensus or a threshold operation, and then release their tiles or their medication to treat a disease immediately. But there are all kinds of other novel contributions of those types of networks. For example, they can use fingerprinting by detecting the DNA of specific tissues via DNA markers and thus compute where they are in the human body, which is also an unsolved problem as of right now. Also, they might be used as an alternative DNA detection mechanism in a petri dish, for example, and is a suitable competitor for PCR detection mechanisms as we used them in the early phases during this pandemic, for example. Thus, you can program those DNA-based nanonetworks almost arbitrarily and detect any kind of DNA. And thus, it is a quite, arbitrarily programmable detection mechanism for DNA that is maybe even faster than the PCR. And it is quite error resistant, as we can encode a lot of conditions for the full assembly of such a message and can thus pretty much arbitrarily choose an error we are willing to take. Also, the entire process is biocompatible by nature, as DNA occurs everywhere in the human body. And we can even use multiple parameters as a diagnosis if possible. Many other fast tests can only detect one marker type, for example, and with DNA-based nanonetworks we can detect multiple markers at once, can compute them in a message molecule, and then react upon the detection. And the best part is we can create those from already existing materials, and there are no such limitations as with electromagnetic communication, for example, where we just have to imagine that those things work. Okay, and in summary, what do DNA-based networks achieve? Well, they are a suitable material for construction, computation, and communication, and all those things can be combined into a working whole, which is fully unclear with most current technologies. It is even possible to create fully functional nanonetworks just from DNA as a building block. And it is possible to compute any Boolean formula at the nanoscale via message molecules in a somewhat feasible manner. And DNA-based nanonetworks only rely on already existing available technologies, and they can be used to perform complex and arbitrary computations, which is a major bonus, which is not already possible at the nanoscale. Okay, but that is pretty much it for today. I thank you very much for your attention, and I hope I sparked some interest into this very interesting technology of nanonetworks as a whole, and specifically of DNA-based nanonetworks. Those are my references, and if you want to contact me or my Professor Dr. Stefan Fischer, you can use the email addresses presented in this contact slide. Okay, but until then, I wish you a nice day and goodbye.